Welcome to G Fun Facts Online, where we dive deep into the science that shapes our world. Today, we're tackling a microscopic menace: viruses. Specifically, we're exploring the fascinating and ever-evolving battle to stop them in their tracks. It's a high-stakes game of cat and mouse, and scientists are constantly developing new strategies to outsmart these tiny invaders. The core of the fight against viral infections really comes down to one thing: stopping viruses from making copies of themselves. Think of it like a factory. If you can shut down the assembly line, you can stop production. To do this effectively, scientists need a deep understanding of how a virus operates, its entire life cycle. From the moment it sneaks into a host cell to when it releases a flood of new viral particles, every single step is a potential weak point, a target for an antiviral drug. One of the main battlegrounds is the virus' own toolkit, specifically its enzymes. These are proteins that act like tiny machines, carrying out essential tasks for the virus. Take viral polymerases, for example. These are the enzymes responsible for copying the virus' genetic material, its DNA or RNA. Without these copies, the virus can't replicate. Drugs like remdesivir, favipiravir, and molnupiravir are designed to jam these polymerase machines, effectively hitting the stop button on viral genome replication. Then there are viral proteases. After a virus makes large strands of proteins, these proteases act like molecular scissors, cutting them into smaller, functional pieces needed to build new virus particles. If you can block these proteases using inhibitors like those found in Paxlovid, or historically, lopinavir/ritonavir, though its effectiveness against SARS-CoV-2 specifically has been debated, you can prevent the virus from assembling itself properly. It's like taking away the construction crew's tools. For some viruses, like HIV, which are retroviruses, there's another crucial enzyme called integrase. This enzyme helps the virus insert its genetic blueprint into our own DNA. Integrase inhibitors do exactly what their name says. They block this integration, stopping the virus from making a permanent home in our cells. But the hunt for targets doesn't stop with these well-known enzymes. Researchers are constantly looking at other viral components. For instance, the influenza virus has a protein called M2, an ion channel that helps the virus uncode itself once inside our cells. This was a target for a while, but viruses are sneaky, and resistance emerged. This led to a shift in focus towards other strategies for flu, like neuraminidase inhibitors, which cleverly block the newly formed viruses from escaping the cell to infect others. Scientists are also looking at non-structural proteins, or NSPs. These are proteins that are vital for the virus life cycle but aren't actually part of the final virus particle. Think of them as temporary scaffolding or specialized machinery needed during construction, but not part of the finished building. Hepatitis C virus, for example, has NSPs that are excellent drug targets. Even the virus structural components, like the capsid proteins that form its protective shell, or the ribonucleoprotein complexes they create, are being investigated as potential Achilles heels. Now here's a major hurdle. Viruses are masters of evolution. They replicate quickly and make mistakes while copying their genetic material. Sometimes these mistakes or mutations can make them resistant to our drugs. This rapid evolution leading to drug resistance is a constant challenge. So how do we fight back against a moving target? One clever approach is to stop targeting the virus directly and instead focus on the host cellular proteins or pathways that viruses hijack for their own replication. Think about it. If a virus relies on one of our cellular machines to operate, and we can temporarily disable that machine without harming ourselves too much, the virus is stuck. This could reduce the chances of the virus developing resistance because it would need to adapt to changes in us, the host, which is a much more complex evolutionary hurdle for the virus to overcome. And this is where cutting-edge technology really shines. We're seeing the rise of nanomedicine, using tiny nanoparticles to deliver antiviral drugs more effectively, improving their stability, and making sure they get to where they're needed. Then there's gene editing. Technologies like CRISPR are being investigated for their potential to directly target and snip out or disable viral genomes, or essential viral genes within our cells. Imagine molecular scissors that can specifically find and cut the virus instruction manual, Programmable antivirals are also on the horizon. Locked nucleic acids, or LNAs, are a great example. These are specially designed molecules that can bind very tightly to critical RNA structures in a virus, essentially gumming up its works. These are showing a lot of promise in early studies. And let's not forget the power of computers. Computational tools and artificial intelligence are revolutionizing drug discovery. Scientists can now model viral protein structures in incredible detail and predict how different drugs might interact with them, even as these proteins mutate and change shape. This massively speeds up the process of finding new drug candidates. The ultimate goal for many researchers is the development of broad-spectrum antivirals, drugs that are effective against multiple types of viruses, not just one. This is incredibly important, especially as we think about preparing for future, unknown viral threats. If we have drugs that target conserved mechanisms shared across many different virus families, we're in a much better position to respond quickly. Another practical strategy is drug repurposing. This involves looking at existing drugs, already approved for other conditions, to see if they might also have antiviral properties. It's a way to find new treatments much faster, because these drugs have already gone through safety testing. Finally, combination therapies are becoming increasingly important. This means using multiple drugs at the same time, each potentially targeting a different viral mechanism, or even targeting both the virus and host factors. This multi-pronged attack can be much more effective than a single drug, and significantly reduces the chances of the virus developing resistance. It's like fighting an enemy on multiple fronts simultaneously. So the landscape of antiviral therapy is incredibly dynamic. We're moving towards more targeted, adaptable, and robust strategies. From understanding the intricate details of viral replication to leveraging AI and gene editing, the fight to combat the ever-present threat of viral diseases is a testament to scientific ingenuity. And that's a G fun fact for today. Thanks for tuning in.